Good afternoon, everyone. It's a real pleasure to be able to welcome Dr. Tottenham, who's with us today for a seminar. Dr. Tottenham got her PhD with Dr. Gunnar and Dr. Nelson at the University of Minnesota, both of them being real pioneers in the field of child psychology and development and stressors, early stressors. And then she went on to a position at UCLA in the Department of Psychology. And now she's a full professor at Columbia, also in the Columbia University in the Department of Psychology. Dr. Tottenham is well recognized in the field of developmental psychology, development of brain in the infants in relationship with early life experiences. She has a number of seminal papers and made a tremendous strides towards identifying the connectivity changes and the factors that are important in determining the effect of early life experience on the brain. She's a member of the Dana Alliance for Brain Initiative. She's a fellow of the Association for Psychological Sciences, and she's an awardee from the National Academy of Science. Dr. Tottenham has been very well funded throughout her career through NIMH, NSF, and other fundings. She has a number of very seminal publications in Neuroscience, biological psychiatry, nature communication. And really, she's coming from a lot of different angles to assess the impacts of early life adversity. So for us, it's a real pleasure and an honor to welcome you here, and we're very happy to hear your talk. So I'm going to leave it to you now. Thank you.
Thank you, Dominique, for such a warm welcome. And also thank you for the invitation to come and speak to everyone at this institute. So I'm going to try and share my screen, and please let me know if I am unsuccessful in doing that. So like many of you at this institute, our research has really been trying to better understand this enduring link between early experiences, especially early caregiving experiences, and later emotional functioning. And the emotional functioning that we've been most interested in are things like emotional learning and emotion regulation. And
excuse me, I'm sorry. Can I ask everyone to go mute? Please. Thank you. I'm sorry, Nim.
That's okay. We're all flexible, right? And so a lot of focus in our lab and also in this talk will be on considering the development of the connections between the amygdala, this subcortical set of nuclei that are helpful in emotional attention and facilitating emotional learning, and its bidirectional connections with the medial prefrontal cortex. And so the argument made by many is that the link between early caregiving experiences and later emotional functioning is at least in part mediated by the development of these connections. But certainly when it comes to humans, this is true for many species, but certainly for humans, when we understand the development of the amygdala and its connections with prefrontal cortex, we appreciate that this development is happening, it's occurring within a very special environment, within the context of a species expected stimulus, which is the parent. And certainly this is universal for humans, and for the purposes of this talk, anytime I refer to parents, in the case of humans, I'm referring to mothers, to fathers, to biological and adoptive parents, to grandparents, or any adult who is the primary caregiver of a child. And what is perhaps most noteworthy about humans is the duration, the amount of time that we spend with our parents, which is on the order of years, which is metabolically very costly to parents to raise another human being. But in exchange, what we get back as a species is this really prolonged childhood, this really prolonged period of brain development that affords us this extended period of plasticity. So in other words, by spending so much time with our parents, parents can provide a sort of scaffolding for our central nervous system development, which affords us this very long period of plasticity. But of course, plasticity is a double-edged sword. It itself is neither good nor bad. It just allows brain development to change as a consequence of the environment. And so on the negative side, this extended period of plasticity may increase the risk for environmentally induced emotion regulation, mental health difficulties. So I'm listing here a number of mental illnesses associated with emotion regulation broadly defined, and age on the x-axis, the red indicating the peak age of disorder onset. So you can see that these are developmental disorders that may be a consequence of our prolonged plasticity. On the positive side, this plasticity affords us great epistemic growth. So it allows us to become the very sophisticated creatures that we are by the time we're adults. So let's start off looking at what we know about human amygdala development, and I'll start with some structural data. Several labs have identified that there is very robust and rapid structural development in terms of volume of the amygdala right after birth. So I'm plotting here data from John Gilmore's group showing that from birth to the first birthday, there's 105% change in volume. And there is continued volumetric growth even through puberty, but those changes tend to be dwarfed by this massive change that we're seeing within the first year of life. And so much of the work in our lab has been asking, what are the functional changes that are resting upon this early structural development? So I'd like to start with an early paper from our lab that brought in, this was a cross-sectional study, brought in individuals between the ages of 4 and 22 years old. And we presented them static fear faces, and there are pros and cons to using these types of stimuli, which I'm happy to talk about afterwards. But suffice it to say that it is a relatively innocuous way for us to engage amygdala activity in children as young as 4 years old. And we were just curious in asking, what should the developmental path look like? What should the shape of the developmental growth look like across the first 2 decades of life? And so we presented these types of stimuli to individuals at 500 milliseconds apiece. And we performed standard pre-processing on each person's brain and then smushed all the brains together to look for regions that were changing in activity as a function of age. And one of the main regions that popped out from this whole brain analysis was the right amygdala. And what I'm plotting here in blue is the correlation between the amygdala signal or the bold signal and age. And blue here would indicate a negative correlation. So to make that clearer, I'll pull out those data and plot them here. On the y-axis is age in years. On the did I say that? The x-axis is age in years. Y is the amygdala bold signal, and each dot represents a participant. And what you can readily see is that the amygdala bold signal to these fear faces tends to be strongest at youngest ages and then attenuates with increasing age. So with these, so what we're seeing is that there's the strong amygdala response early in life, which makes a good bit of sense when you're younger and when you're relatively new to this world. You have a lot more learning to do. So this is really consonant with the amygdala's role in emotional learning. But we can use these same data to measure functional connectivity, which I know many of you are experts in. But for those of you who are not, functional connectivity briefly involves extracting the time course signal or the bold signal from one region like the amygdala and plotting it over time, and doing the same for another region like the medial prefrontal cortex. And the degree to which these two time courses are correlated with each other gives us an index of functional connectivity. So it's essentially suggesting that there's some conversation that must be happening between these two regions. So using fMRI data, what we and many others have seen is that there are large changes in amygdala connectivity with prefrontal cortex from childhood to adulthood. So what I'm showing you here now is not activity, but it's connectivity between the amygdala, which you can't see here in this slice, and the medial prefrontal cortex. And what people find is in adulthood, there's strong functional connectivity as well as structural connectivity between amygdala and prefrontal cortex. But we don't see that same strong connectivity during childhood. So we get slow development of these connections with prefrontal cortex. So putting these data together, what we're seeing is that this is age on the x-axis. We get a strong amygdala response to environmental stimuli early in childhood, but that's occurring in the absence or the relative absence of these adult-like connections with medial prefrontal cortex, which we don't see emerging in our hands. We don't see emerging until the onset of adolescence. So that's made childhood a very interesting time for us to think about in terms of whether this is a sort of biological substrate that facilitates the environment having a larger impact in the way that the system constructs itself. Moreover, I'll get into this later, but we have some reason or some pre-hypothesis, I'll say, that the strong activity of the amygdala early in life may actually be a biological prerequisite for the instantiation of those adult-like connections with prefrontal cortex. Okay, so if this period is more amenable to environmental change, then we want to be thinking about early stressors that are happening during this time and how those early stressors might be impacting the construction of the system. So if we want to focus on stress, there's obviously decades of basic neuroscience research to turn to, and I just plot here two examples showing that these are data from adult male rodents who are experiencing chronic immobilization stress. And often what people find is that this type of stress may lead to hypertrophy and hyperactivity of amygdala neurons. And the opposite might be observed in medial prefrontal cortex neurons. So taken together, what this pattern is suggesting is that both regions are stress sensitive, but they may be occurring in a region-specific manner. So for humans, when we think about early life stress, one of the most potent stressors may be caregiving-related stress. And for much of my career, I have had the privilege of working with families who have adopted children who experienced early institutional care, or what we commonly call orphanage rearing. So this is an environment where the expected parental stimulus is absent. It's a potent stressor for a human infant for a number of reasons. But what I want to point out is that in the samples that we have been able to work with, the early adverse environment A comes to a rather abrupt end at the point of adoption. So the families that we've seen have all experienced adoption. And the families that tend to adopt internationally tend to provide a highly enriching environment. The parents tend to be highly devoted to providing care for a child and so on. So this sample offers a fairly unique opportunity to understand. We have some better temporal resolution about the early adversity that children would have experienced. Now, children who have experienced early institutional care followed by adoption show they're at great developmental risk, but show great rebound in a number of domains upon adoption. And those domains are often in the areas of growth or IQ, cognitive development. But children may remain at elevated risk for emotion regulation difficulties. Although, of course, there are huge individual differences in outcomes that I will be sharing with you. So the samples that we have seen, the majority of children were placed in institutional care between birth and 6 months, and the majority adopted by their second birthday. So in other words, this experience, the early institutional care experience, was largely occurring before the onset of explicit memory. And so we began by looking at amygdala volume, and this was an accelerated longitudinal study. In red are the PI, or previous institutional care group, and blue is a comparison group that never experienced separation from parents or institutional care. And what we observed early on, this is age on the x-axis, what we observed early on is a slight increase in the PI group in amygdala volume relative to the comparison group. But that slightly larger volume early on was followed by what looked like a slower growth over time. So much so that by the time we reach adolescence, we see that the PI group now has a smaller volume of bilateral amygdala relative to the comparison. And I point this out because I think that what we have been compelled by is that these adversity or stress-related phenotypes themselves develop. So the pattern of results that we get should greatly differ by the age at which we're asking the question. To investigate the functional significance of amygdala phenotypes within this group, we have administered a face go no go task, which was first developed by BJ Casey. And in this task, very briefly, it's a child-friendly task that asks children to press a button or go every time they see a certain face, let's say a happy face. But once in a while, they will see a fearful face and they should not go. They should withhold pressing. And so this is a very hard task for children to do because they build up the prepotent response to go, but it allows us to measure behavioral regulation in the context of affective information. And what we observed was when faces had positive affect, there was no group difference in accuracy between the comparison group and the previous institutional care group. However, when the faces had negative affect, everyone's accuracy dropped, but this is where we saw the group differences, such that the PI group had a significantly harder time regulating behavior in the context of those highly arousing stimuli, which is consonant with parent reports of behavioral difficulties in the context of emotionally arousing information. While children perform this task, we also had the opportunity to collect scanning data. And we observed, and this is now in two separate cohorts, we observed hyperactivity in the amygdala in response to those fear faces following early institutional care. And we observed altered functional connectivity between the amygdala and the medial prefrontal cortex. And the nature of this alteration that we observed was, although this effect needs to be replicated many times, suggested that the children in the PI group may actually be exhibiting more adult-like phenotypes. And I'll explain how that played out functionally. But there are animal studies, some of them coming from researchers within this institute, suggesting that some of this accelerated development may be observed across species and may be a type of adult-like phenotype that's observed within these emotion regulation circuits. So at a behavioral level, what we observed was that when we look at anxiety, the post-institutionalized group overall had higher anxiety. In this case, I'm plotting separation anxiety. But for those children in the PI group who had the more adult-like phenotype, and I'm using adult-like because I can't be confident that this is a more mature pattern. It's just consistent with a more mature brain pattern. But that more adult-like phenotype did seem to be helpful in terms of anxiety in reducing anxiety currently at the time of assessment relative to those PI children who did not show that phenotype. However, we've been able to track these individuals across the next few years. And I'm going to, in the next slide, replot these two bars here to show you what happens over time. So those are the same two plots. In red are the PI children's anxiety with the adult phenotype in the brain, and blue, those without. And the numbers are small here, so we still have a lot more work to do here. So I just want to caution, there's, I think, only about 40 children in each group. But in 2 years and then again in 4 years, we see that that rank ordering switches such that the children who showed the more adult-like phenotype in the long run may have retained some of that higher anxiety, whereas those children who showed the more childlike phenotype at the first scan were more likely to drop in anxiety scores over in the long term. So perhaps what's happening is in the context of amygdala hyperactivity that was the result of an early caregiving related adversity, that hyperactivity of the amygdala may have instantiated the start of those connections, those adult-like connections with medial prefrontal cortex at an earlier age. However, if that's true, then that might also be accompanied by a reduction in developmental plasticity that we might see in the absence of that type of early caregiving adversity. So these data and those of many others suggest that the links between early caregiving experiences and later emotional functioning is mediated in part by the development of connections between amygdala and prefrontal cortex. But the question that's really been of great interest to me lately is asking the question, how is this happening, or why is this happening? And that's turned me to some more fundamental research on what parents are good for. And I just told Dominique that I am coming off of sabbatical, so I did a lot of reading, and some of these papers are older, as you'll see, but they're really wonderful. And I wanted to share them in this talk. So during development, humans have a very strong drive for epistemic growth and exploration. But that exploration comes along with great risk to the child. And so our drive for epistemic growth has to be kept in check with our need for security or safety. And so if we take, for example, this child who's exploring his environment, he's able to do so because of this species expected stimulus. The parent is able to provide either a secure base or safe haven that affords this child the luxury to explore his environment. And so as Bischoff has told us, as children explore the environment and encounter something new, one of the things that may happen is arousal increases. And arousal itself is it's not valenced, but a heightened arousal can lead to either an approach or a preference for a stimulus, or it can lead to an avoidance of a stimulus. And that line between approach or avoid, or as I observed in my children, that line between fear and fascination can be very thin. And I included here a picture of my own daughters approaching a bug that they had never seen before. And my memory of this was this wavering back and forth between, I want to approach the bug, I'm scared of the bug. And what Bischoff describes is that decision point between approach and avoid following increased arousal is the affective context. And parents and the relationship with parents is certainly going to be a very large source of affective context. So as Strofe and Waters have told us, the attachment relationship is an asset to offspring. It facilitates the pursuit of children's future social and exploratory goals. So to demonstrate, this is another older paper showing the infant's responses to a stranger's approach. So on the y-axis is age and months. X-axis is age and months. Y-axis is the infant's response. Positive is high, negative is low. And the red line are infants who were left, their mother is gone, and their father, and they're left with a friendly babysitter. And what you can see is from 8 months and earlier, infants overall approach the stranger with positivity, but there's this sharp drop off around 9 months where they start to show more wariness and perhaps even fear to the stranger. When the infant has its mother present, you can see they have that positivity early on, perhaps even more so than when the mother was absent. But the mother's presence attenuates that sharp drop off at around 9 months, such that the mother's presence is keeping the infant's affect at around a neutral state. That neutralness is misleading because what's not plotted here is a lot of individual differences, with some infants expressing positivity and others expressing negativity. So these data make me think about the way that I often historically thought about approach and avoid as, being polar opposites of each other and living in different universes in a way, and made me more appreciate that during development, at least, approach and avoid behaviors may live in closer proximity than I had previously thought about. And indeed, Bischoff has suggested that in order to understand the development of avoidance behaviors or fear behaviors, we must also understand the development of affiliative behaviors in humans. So when we think about, when we look at altricial species, and I include humans here in that they are far from adult-like, there's often observed a maturational delay between affiliative or security systems and fear-related systems. And that delay or that lag, it's argued, has to exist so that the affiliation or the attachment with the primary caregiver can be established without fear learning systems interfering. And then later, once fear systems start to develop, those attachment relationships that were formed earlier can facilitate management of fear encounters in the future. So to demonstrate, here are some puppies. So the top puppy was exposed to a human handler during an affiliative critical period, whereas the puppy on the bottom was first exposed after this affiliative critical period. And the data are plotted here. On the x-axis is the age of first exposure to the human handler, and the y-axis is the puppy's attraction score. And what you can see is initial exposures that are occurring around 5 to 6 weeks produce an affiliation. First exposures that occur later on produce a fear or a wariness. And what's interesting is that this affiliation learning occurs regardless of the valence of the handler. Both friendly and cruel handlers will produce an affiliation during this stage. In fact, some studies have shown that affiliative learning is actually increased by shock administration. And perhaps it's for these reasons that Bowlby indicated that these early affiliative learning processes seem to defy the rules of reinforcement learning in that they occur regardless of valence. So following this initial attachment formation, Several, I'm listing here Regina Sullivan's work, several have shown that the attachment figure, once established, can then serve to facilitate future approach or preferences of novel cues. And so for those of you unfamiliar, in Regina Sullivan's lab, what she's shown is if she were to pair a neutral stimulus like a peppermint odor with a foot shock, under some conditions, the animal, and then you place the peppermint odor in a Y maze on one arm. In some conditions, the animal would avoid the peppermint odor, as you and I might do. But there are other conditions where the young rat pup might approach that peppermint odor. Now, when animals avoid the peppermint odor, that is amygdala mediated. But if the amygdala is quieted and prevented from mediating this learning, that's when approach behaviors are observed. And so to better illustrate this, what Sullivan's lab has shown is that if this is pup age on the x-axis and amygdala modulation on the y-axis, there's this period postnatally when the functioning of the amygdala during this Pavlovian fear conditioning is dependent on the presence or absence of the mother. So when the mother is present, her physical presence is keeping corp production low, and amygdala does not engage in fear learning. In that same animal, if you remove the mother from the nest, then those processes reverse and amygdala engagement becomes possible during fear learning. And so the animal acts as you and I would, avoiding the fear stimulus. So in other words, when the animal is conditioned in the absence of its mother, it will subsequently avoid the peppermint odor as you and I would, and that's what's being plotted here. But if this same rat pup were conditioned in its mother's presence, subsequently this animal will approach the peppermint odor because the amygdala was in essence buffered by the mother's presence. And that's what's being shown here, the greater number of approach behaviors when the animal was conditioned in the mother's presence. So we are at the earliest stages of asking whether humans do something similar, but it seems to make sense that they should, given these fundamental affiliative learning processes. So we collaborated with the Sullivan Lab, and we invited young preschool age children to the lab and presented them with a conditioning study, the conditioning paradigm. A blue rectangle was paired with not a shock, but we used an aversive noise, like a loud sound of nails scratching down a chalkboard. That was the CS plus. The CS minus was never paired with that US. And children either learned this association alone, or they learned it with their parent present, and their parent wasn't doing anything, they were just physically present in the room. Following this conditioning, parents went away, and we placed children into a human Y maze, which was a tent that children would enter. It was a play tent, and the CS was on one arm, CS plus on one arm, CS minus on the other. And we asked children to enter the tent and choose a door, right or left, to retrieve a prize. And we showed them that it was the same bucket of prizes behind both doors. We just wanted them to pick a door, and they did that 5 times. Okay, so first I'm going to show you the data during acquisition, and it's a very messy slide that I will be sure to walk you through. So we had 4 metrics of learning during acquisition. We had a reaction time measure, accuracy, we measured children's heart rate and their skin conductance. And if you can ignore the differences between blue and gray and just pay attention to the CS plus versus the CS minus, what we found was by all 4 metrics, these very young children were able to distinguish the CS plus from the CS minus. Now, if you pay attention to the difference between the blue and the gray data, what we also observed is that parents were not having a significant effect on these initial acquisition measures. So what this suggests to us is that parents weren't simply calming, if we're going to see any effect, parents weren't simply calming children physiologically. So now if we look at the data, the recall data when children were placed into the Y-maze tent, we see that when children conditioned alone, that's the top image, it's a modest effect, but these are children crawling through a tent. So we were happy to see a modest effect. Children were less likely to approach that CS. In other words, they were more likely to avoid that CS plus, as you and I would. However, when children conditioned in the presence of their parents, they were statistically more likely to approach or prefer that CS plus. So in other words, at least at the level of patterning, children were performing in a similar way to the rat pups in that their learning was affected by whether their parent was present or not. And we found that this effect of the parent was associated with children's court production in the lab. So children who produced higher levels of court were less likely to be affected by their parent being present in the room or not. They were more likely to avoid that CS plus, just like you and I would, regardless of whether their parent was there, suggesting that there might be some biological constraints on this learning. And finally, we used what's called a dot probe task. And if you're not familiar with this, it's a very simple task where we place the CS plus and the CS minus side by side on a screen. And on the next screen, children saw an asterisk, either on the right or the left. And all they had to do was indicate whether the asterisk was on the right or the left side of the screen. And the nice thing about this task is their speed, the speed with which they press the button, gives us an indication of whether their attention on the previous slide was more on the right or the left side of the screen. So using this type of probe, we found that when children conditioned alone, they were more likely to avert their gaze away from the CS plus, consistent with what some other studies have shown, that children will avert their attention to fearful stimuli. But when children were conditioned in the presence of their parent, their attention was biased towards the CS plus. What I didn't mention, I forgot to mention, is that this was using a within subjects design, meaning that these are the same children whose behavior and learning is being modulated by their presence, their parents' presence or absence. So at a behavioral level, I can't say that it's exactly the same thing, but at a behavioral level, we're seeing similar patterns to what has been identified in rodents. We also wanted to understand whether parents were affecting amygdala function and brought children into the scanner. This was actually children and adolescents. And we presented them with images of their parents versus images of somebody else's parent, in other words, a stranger. So we were just showing them in a block design, parents versus non-parents. And what we observed is that in childhood, the amygdala was showing a decreased response to the parent relative to the non-parent, which we interpreted as parental buffering of the amygdala. The parental cue was leading to a decrease in amygdala signal during childhood, but we didn't see that same modulation in adolescence, suggesting that this parental effect, if replicated, starts to attenuate as children enter adolescence. And we observed that the presentation of the parental cue was strengthening, phasically strengthening the connectivity between amygdala and medial prefrontal cortex. So all of these effects so far depend on the parent being regulated, for lack of a better term. The parent is not expressing fear. The parent is just present and not reacting in any particular way. But we know from many rodent studies that the parents' affective cues are potent amplifiers of the offspring's stress reactivity. And so we performed this study where we flipped things around. Here, we were interested in the parent transmitting learned fear to the child. And so instead of fear conditioning the child, we fear condition the parent and videotape the parent and showed the videotape to the children. So here's me getting fear conditioned. Every time I saw this pink circle, I would flinch because I heard that horrible noise. And here's another adult doing the same thing, but learning that the green diamond is aversive. And so my daughter would watch both of these videos, and then later we would test her responses to the cues themselves. And what we found was after acquisition, the amygdala, children's amygdalas were more reactive to the cues that their parent was conditioned to relative to the cues that an unfamiliar parent was conditioned to. And while children learned to dislike both stimuli, both CS pluses, they learned better to dislike from their own parents. So to better illustrate all of these findings together, I've adapted Wundt's curve of hedonic function. And this curve works like this, that as arousal increases from low to mid-range, individuals tend to be more likely to show approach-related behaviors, or what I'm calling here a fascinated state, an interested state. If arousal gets too high, then individuals will switch over into a more avoidance state or even fear. And so perhaps what parents, the presence of a regulated parent is doing is to help maintain children in this approach fascinated state. But if a parent themselves is distressed or even frightening or just absent, then perhaps children are more likely to shift into this more avoid or frightened state. And so we returned to our parent stranger task in the scanner. These are the data I just showed you earlier, where children with a typical caregiving history showed what we are calling this parental buffering of the amygdala. And now we included children who experienced early institutional care, meaning that they experienced early emotional learning in the absence of that primary caregiver availability. And here, we did not see that same buffering of the amygdala by the parent, suggesting that at a group level, this early experience was placing limits on the amygdala's ability to be buffered by this parental stimulus. But I want to be quick to point out that these are very large error bars here, which indicates that even within the post-institutional group, there were children who showed buffering to their parent, this is their adoptive, to their parents' image, and those who didn't. So we interrogated these individual differences longitudinally, and I'm plotting here anxiety on the y-axis. And we brought all of these participants back 2 years later to measure their anxiety. We split participants into those who showed buffering at the level of the amygdala, and I had buffering in quotes, and those who didn't at time 1. And if you pay attention to the right side of the screen, the previous institutional care group, we see both those who showed buffering and those who didn't at time 1 did not differ in terms of anxiety. But 2 years later, when brought back, those who showed buffering to their parents' image at time 1 showed significant drops in their trait anxiety. And those who did not show the buffering were likely to maintain those elevated levels of anxiety. So perhaps like us, you're curious as to what differentiated these two groups. And in this case, it was not the age of adoption, which is a variable that often comes up as having great explanatory power. That didn't come up in this case. Instead, what we found was those children with a history of institutional care who showed this buffering at time 1 were also those who reported stronger felt security with their parents, or a middle childhood proxy for attachment. So it suggests that the relationship with the parent was distinguishing these two groups of children. And that's consistent with other findings. Again, these are all cross-sectional, but other findings in our lab showing that the greater felt security that children in the post-institutional care group had, the lower their internalizing or anxiety and depression symptoms. So what this suggests is that a feeling of security is always important for all children. It may be especially important for children who have experienced early caregiving related adversities. So, so what we think is if I put this all together, we think that if this is age on the x-axis here, there might be this period during childhood when parents are an especially potent stimulus for increasing or decreasing amygdala and related processes. Maybe parents aren't the only important stimulus, but parents may be a very important stimulus that has a privileged access into limbic development of their children. And the nature of what happens during this time may be consequential for the way that this system continues to operate once this putative sensitive period closes. So I just want to end my talk by sharing with you the next steps that our lab is heading in, some of the questions that are we find exciting, and these are motivated by the recognition of convergence across several separate lines of research. So one is that the amygdala and medial prefrontal cortex circuitry seems to be coming up in a lot of different literature. So one that I just shared with you is that this neurobiology is highly modulated by parental cues, especially early in life. This same neurobiology is also altered or different in children who've experienced early caregiving adversities, and I should say in adults who've experienced early caregiving adversities. The same neurobiology is the neurobiology that often comes up in the literature on emotion regulation processes in healthy adults or unhealthy adults. And finally, the same neurobiology routinely appears in the literature examining how we learn and maintain semantic level emotional knowledge or semantic level affective knowledge. So I have been very compelled by this nice paper by Roy et al., who present a conceptual synthesis of many literatures, providing a parsimonious explanation for a unified neural function of what are seemingly a disparate array of behaviors supported by amygdala and medial prefrontal cortex. It's actually all subcortical circuitry and medial prefrontal cortex. And this broad array includes things like emotion regulation, valuation and decision-making, self versus other processes, mentalizing about others' actions, somatic markers, default mode functions, and so on, that all seem to center on affective meaning representations. They argue that while subcortical regions are needed for basic forms of learning and valuation, perhaps medial prefrontal cortex holds the abstractions from routinized events. In other words, it's holding a more semantic level information than these subcortical regions. So that is the medial prefrontal cortex and its subcortical connections construct affective schemas about how the world works from specific cues, and then facilitates recall from similar past situations to guide prospection about future potential outcomes. So this type of thinking that Roy et al. Have laid out have generated a working hypothesis for us that the emergent patterns of amygdala and prefrontal function that we see reflect, especially after early adversity, reflect the affective semantic memory structures or the schema-based structures that over the course of development construct these emotional schemas that guide predictions about subsequent social and emotional stimuli or contexts. So in other words, what I'm currently thinking is that these deep learning structures like the amygdala are very useful, especially early in life, for episodic like learning about particular events. And in the context of caregiving, it could be simple binding of things like, I have a need and my need is met, or I have a need and my need is not met. So over time and development, and repetition, which is running down the screen, over time, development, and repetition, information from these deep learning structures can be abstracted or semanticized to regions within the medial prefrontal cortex and held in semantic form rather than episodic form. And with increased time, the connections between these subcortical and cortical regions are the representation, or at least part of the representation, of these schematic memory processes that allow us to make meaning from discrete events that we have, in this case, with our caregivers. And these semantic structures are what help us make future predictions about subsequent social or emotional encounters. And if individuals encounter information that greatly violates what's held in these semantic structures, then updating of those semantic structures could theoretically occur through processes of assimilation or accommodation. And presumably that's what, for example, therapy is doing, is to help update these affective structures. So as Alan Srofe has written, the attachment relationship is the organizer of thought and action, at least in the realm of social emotional development, if not more. And these attachment schemas generate predictions about future social and emotional encounters. So perhaps the individual differences that we see in subcortical cortical function in adulthood reflect the learned or semanticized schemas that were constructed from routine early caregiving experiences in a way that allows us to make meaning at a neural level. And so I'm thinking that what we're perhaps seeing when we see differences in people's bold signal in amygdala and medial prefrontal cortex are different abstractions at a neural level regarding trust that's been learned from the environment, whether it's trust in others, trust in myself, trust in the world, and my relationship with others. So this is a line of working, a line that we're pursuing right now. So to conclude, I've explained that the human amygdala and medial prefrontal cortex develops very slowly, but that there may be value in this slow development. This long sensitive period for the environment to influence connections between the amygdala and prefrontal cortex is one of the things that childhood affords us, where the environment can provide rich scaffolding for a long time. And parents, again, they're not the only source, but they're an important source. Parents are a source of social regulation over the development of amygdala and prefrontal cortex, having some perhaps privileged access. And so perhaps, therefore, not surprisingly, early caregiving related adversity is associated with differences in amygdala medial prefrontal development. Perhaps one can expect to see some immediate benefits in some children with some later occurring positive experiences, especially surrounding the family. And finally, perhaps one of the major roles of caregiving is to help construct this emotion regulation neurobiology at the level of schemas that abstract meaning from socio-emotional encounters and help make predictions about future ones. So with that, I have up here the funding sources. I'd like to thank the lab members, past and present, and especially the families and individuals that participated in this research. And thank you for your attention. And I'm happy to take any questions if we have time.
Thank you very much, Nim. That was a fantastic talk. Of course, I'm sure that we could have listened to you for a lot longer than an hour. You have so many data and so many new data also coming out that are really interesting, but I think we already have a few questions. Okay, so I'm seeing Suzanne. Can you speak to the idea? Okay, this is a question from Suzanne King. Okay. Because she has problems getting her microphone to work. Can you speak to the idea that helicopter parents who perhaps provide too much security for their kids might actually be associated with greater anxiety in children?
Yes, thank you, Suzanne. That's a great question. And I think that this framework really lends itself to be able to address this question of, the helicopter parent or the bulldozer parent, because I think if we think about sensitivity of the parent, which attachment theorists have always said is one of the secret ingredients of a feeling of security, that being sensitive to the child is being sensitive to that balance that I started off the talk on about this drive for epistemic growth and autonomy, as well as a drive for security. And so a parent who is serving as a helicopter, as you say, is in some ways depriving a child of those opportunities for exploration and autonomy, which is so critical for development of trust and self also. So I think that this may be part of why there are links between that over-parenting and later difficulties with things like anxiety. And so, yes, so I would say that balance, and that balance theoretically, and I'm certainly not the first to say this, but that balance is going to be different for each child, depending on their own individual differences and to the developmental stage of a child. So you would have different balances for a 2-year-old versus a 17-year-old. But that's a great question. Thank you.
Thank you. Do I see other questions here? I have a question for you. I have 2 questions, in fact, for you. One is you were talking about higher corticosteroid levels or cortisol levels that are associated with lower influence of the parents. So can you maybe just extrapolate or just emphasize what you mean by that, and in which context you've observed that?
Mm-hmm. Yeah. So thank you for giving me the opportunity to expand on that. So some of this is speculation and drawing big lines between faraway dots. But what we have seen, if we look across age from, let's say, 2 years old to adolescence, is we see a steady increase in baseline CORT. And I'll define what I mean by baseline in a second. And so the reason why I showed those CORT data was because reading between the lines, what that's a bad way to say it. What I think if more research could occur that included longitudinal work and a wider age range, I think what we're seeing there is the children with the slightly higher CORT levels may be maturing that system a little bit earlier relative to the children with the lower CORT levels. That's one possibility. And so, in other words, there may be some developmental maturational constraints where you start not being so influenced by your parent within that learning context. The other possibility, which is not orthogonal to that explanation, is that now I have to explain what baseline court is. So these are not truly baseline court data because they were collected 30 minutes after the child arrived into the lab, which is not really baseline. And they're with their parents. So those individual differences may also or only be indexing children's court buffering by their parents in response to coming into the lab. And with our data, we cannot say. That really requires some additional research.
Yeah, thank you very much. I have another question from Suzanne. That's too bad she cannot open her microphone. Thinking about kids who wind up in institutions in infancy, there may well have been a lot of prenatal stress. To what extent do you think some of the effect on the amygdala development might be due to the prenatal environment?
Yes, this is really important. Surely the amygdala, as many have shown, is sensitive to the prenatal environment, and surely the mothers who are in a situation where their child is going to end up in an institution are presumably under stress. So I do think that there's an influence of that prenatal environment that unfortunately we don't have access to know. But I will say, in addition, there is a postnatal contribution in that, well, I will infer that there is an additional postnatal contribution in that in many of our studies, we find an age of adoption correlation with an amygdala phenotype, suggesting it's certainly not conclusive and we can't do the elegant causal studies that animal researchers can, but it's suggesting that there is some kind of dosage effect of staying longer in an institution and having less time in a stable family.
So thank you very much, Nim. If I may, I have another question for you also, is you were mentioning also the change in the timing of maturation of amygdala to prefrontal cortex connection versus the descending connections that are more related to the inhibition of fear or suppressing fear memory. So what do you think that in the case where we have an accelerated maturation of the prefrontal cortex, possibly due to this increased amygdala activity in early adversity, that we might see a similar acceleration of the descending projections? What would you predict for that?
My prediction would be yes, that we would see that. But part of that prediction is that if, and this is all a big if, at least based on the work that we've done, if that is the case, that you've got an accelerated development induced by these early pressures, I don't expect that the resulting phenotype will be the same as an individual who did not experience that acceleration. And I think that it's at a behavioral level, there are concepts of growing up quickly after experiencing adversity. And I think that perhaps at a neural level, maybe we're seeing evidence of growing up quickly at a neural level, and it's to the individual's advantage to grow up quickly in that it helps you meet immediate needs. But it also would therefore come along with a potential cost of truncating that sensitive period and not allowing for the tremendous amount of elaboration that must occur within those emotion regulation circuits. So again, this is all speculation, but I would predict that, at least the way that we talk about it in humans, that an accelerated development might be more likely to result in more reflexive emotion regulation behaviors in adulthood rather than more reflective emotion regulation in adulthood.
and then you would think that the concept of the semanticized schema that are, like triggered or that are facilitated by the early care might be totally different than at this point, even regardless of the physical maturation of those connections then.
That's what we're thinking right now, is that the content will different. The narrative that's represented at a neural level may differ as a consequence of these experiences that we've had early on.
And do you think that, for instance, in children that have been adopted from institution, and do you think there might be intervention that could be carried out to modify this early engraving of the narrative, for instance?
Yeah, so I would say that I think that for children that have been adopted, their families are the largest intervention. That's a huge change in their environment. And again, I went over it very briefly, but the families who adopt internationally, we infer, have a great dedication to providing care for a child, because it's not easy to adopt internationally. And in fact, it's that desire that's been associated with the best outcomes for children. That being said, I think that the way that sensitive periods work, as you know, is that it's easier to instantiate change when you're in a sensitive period, much harder afterwards. But unlike a critical period, the idea with a sensitive period is it's not impossible, right? It's just, it takes more exemplars. And so I think that, in my last slide, I was suggesting that if there's information that is violating enough of your existing schemas, that perhaps that can override the way that these early structures are set up. And I think in the data that we see, where children who have a strong sense of security with their parents, again, I can't talk about directionality because these are correlations, but I think that is speaking to this idea that for those individuals, there may have been experiences potent enough to override some early learned lessons.
Mm-hmm. That's at least that's what we wish for.
Mm-hmm.
All the time. So thank you so much, Nim. I'm just going to look if there are other questions from the audience. I don't see any, unless I'm mistaken. But if there are no other questions from the audience, then
I think there is another question on the chat.
Oh, okay. So, yes.
Oh, I see it.
Okay. So, as a social species, this is from Vanessa Oliveira. As a social species, we are born very immature, but ready to absorb human culture, as unfortunately, it does not come out of our genes. And that is done with the expected parent species stimulus, while our amygdala is very reactive and on the loose.
That's true. I think that, that is the double-edged sword in a way of plasticity, is just that I think I don't want to, assume I understand Mother Nature too much, but I think that is right, that we're born with our amygdala, as you say, Vanessa, on the loose. But in general, that's a good thing, because it helps us learn from those around us, right? I mean, that's the wonderful thing about being a social species, is that you don't have to do firsthand learning. You can learn from those around you what mushroom to eat or what mushroom to avoid, how to act around elders in your community, and so on. And so I think that, Mother Nature did it right in that she made these early emotional learning systems ready to go to learn from those around us.
Thank you. Oh, I'm your fan, right? We are all your fan. Thank you very much for your talk. So yeah,
thank you.
Really need to thank you so much for your generosity in giving the talk and answering the questions. I think it's a fascinating area of research, and you've certainly shown a lot of very important aspect to it, and will continue in the future. So we're very looking forward to your research coming out. So thank you again.
Thank you, Dominique.
Thank you.
Thank you. I hope you all
Thank
stay
you.
well. Mhm.
Yes, you too.
Thank you. Bye-bye.
Bye-bye.